Right, class. So let's talk about nucleic acid. As I told you, certain important things that how basically the invention of genetic material took place, and in the same context, I need to describe also many things. I mean, related to this. So first of all, regarding the time frames that I have discussed in the class also, and in that regards, I have already told you that what is the feature of nucleic acid. I mean, why you call it as a nucleic acid, because actually it was discovered from a nucleus, and initially it was discovered by Frederick Miescher. That's why people used it, named it nucleic acid later on. But word nuclein was initially invented. So when I'm talking about the time frame, so one is in 1800s, and then I told you about the name of Frederick Griffith. Frederick Griffith was the scientist who has actually carried out the experiment on transformation. What was that experiment? See, it was very simple that Griffith has actually started his experiment with strains of pneumonia, streptococcus pneumonia actually. So streptococcus is nothing, it is a chain of cocci bacteria, and he has chosen, actually he has found out that there were two different strains. One is S strain, one was R strain. S represent smooth strain and smooth variant, and R is the rough variant. S strain is actually the virulent strain, and R strain is the non-virulent strain. If you see S strain over here, it looks very smooth onto the colony form onto the plate, while if you see R, you see it is a very irregular kind of strain. Okay, so that's the difference between R and S strain, nothing new. Again, I would be repeating, S is smooth, which is virulent strain, which has the ability to cause the disease. R is the rough strain, which is non-virulent strain that do not possesses the ability to cause the disease. It is somewhat irregular onto the plate. Okay. So what Griffith has done, Griffith has chosen to perform certain experiments. In the first experiment he has performed by choosing S strain and injecting it into the mice. Once he injected that thing into the mice, he observed that the mice died. Then in the second experiment he has chosen R strain like this, and he has injected the mice, and what he has observed, the mice remain alive. In the third set, he has chosen the same S strain, but he heat killed it. Now after he chose that S strain, when he heat killed it, he injected that into the mice. He observed that mice survived. So the same S strain which causes the disease earlier, now when the same S strain was heat killed, he saw that after injecting this into the mice survived. I mean, the disease causing ability of S strain has been lost. Then in the fourth set of experiment, he has chosen the heat killed S strain and mixed it together with the alive R strain. Please mark my word, heat killed S strain and live R strain mixing together. ऐसा नहीं करना कि हीट किल्ड आई मीन एस स्ट्रेन और आर स्ट्रेन को मिक्स किया और फिर हीट किल्ड किया. नो, हीट किल्ड एस स्ट्रेन एंड अलाइव आर स्ट्रेन, है ना. तो प्रीवियसली हीट किल्ड एस स्ट्रेन and alive R strain. So he mixed themselves together and he injected the mice, and he observed that mice died. See, the point is that the same R strain which was unable to cause the disease earlier has now somewhat is now able to cause the disease, right? So the heat killed S strain which was unable to cause the disease is now, when mixed together with alive R strain, they proposed certain changes, and this means that there was something. And what was that? Actually, that R strain which was unable to cause the disease earlier, in presence of heat killed S strain, has taken certain factor, that factor which was able to cause the disease, and it was present in S strain, and it actually does not get destroyed by heat killing. It is the phenomena applied over here. It is called as transformation. So the R strain got transformed, right? So that is the main key feature over here. So the word transformation was first coined by Griffith. So the next alarming question is, what is this transforming principle? I'll upload in my next slides.